Welcome, everyone. Today, we are going to explore the salient features of Protochordata. If you have ever wondered how complex animals, including humans, evolved from simple, brainless organisms, you are in the right place. This topic is foundational for understanding animal diversity, and by the end of this video, you will see exactly why these tiny marine creatures are so incredibly important to our own evolutionary history. Let us start with a simple analogy. Imagine you are looking at the blueprints for a highly advanced modern skyscraper. That skyscraper represents complex animals like birds, fish, and humans. But before engineers could build that massive structure, they had to build simpler, smaller prototypes to test the core structural pillars. In the animal kingdom, protochordates are exactly those early prototypes. They are small, simple, ocean-dwelling creatures. They do not have a hard, bony skeleton, and they do not even have a true brain. However, they possess the very first biological blueprints for a backbone and a spinal cord. By studying them, we are literally looking back in time. Now that we understand what they represent, let us define them formally. Protochordata is an informal category of animals within the broader phylum Chordata. You will often hear them referred to as acraniata. The prefix a means without, and cranium refers to the skull, which perfectly describes them because they completely lack a true skull. Furthermore, they do not have a vertebral column or a true backbone. Despite lacking these bony structures, they are exclusively marine animals. You will typically find them burrowing in the sand or attaching themselves to rocks in shallow coastal waters, quietly filtering their food from the ocean. Even without a skull or bones, why are they classified with us chordates? This brings us to their most important feature. At some stage in their life cycle, every protochordate exhibits 4 hallmark characteristics. First is the notochord, a flexible rod that provides essential structural support. Second is a dorsal hollow nerve cord, which acts as a primitive spinal cord to transmit signals. Third, they have pharyngeal gill slits, which they use primarily for filter feeding. And fourth, they possess a postanal tail that helps with propulsion through the water. Every single vertebrate, including you and me, shares these 4 fundamental traits during embryonic development. Before we move on, let us see this with a concrete visual example. Here is a schematic diagram of a typical protochordate. Starting on the left, you can see the mouth where water enters. Just behind the mouth are the pharyngeal gill slits. Notice the arrows showing water passing through them for filter feeding. Now, look along the top of the animal. The long, continuous rod is the notochord, providing that crucial, flexible support. Right above it sits the dorsal hollow nerve cord. Finally, at the far right, you can see the postanal tail extending past the digestive tract. This simple body plan is the foundation of all vertebrate anatomy. Having seen their anatomy, let us look at how this group is divided. Protochordates are primarily split into 2 subphyla based on their notochord. The first is Urochordata. In these animals, the notochord is only present in the tail during their larval stage. Once they grow into adults, like the sea squirts attached to coral reefs, they lose it. The second subphylum is Cephalochordata. Here, the notochord extends all the way from the head to the tail, and importantly, it persists throughout their entire life. The landslits, which burrow into coastal sand, are the classic example of this group. Both groups use the same basic survival strategies, just with slightly different body plans. You might wonder why we care so much about these tiny, brainless creatures. Their importance lies in their immense evolutionary significance. They act as the crucial missing link between simple invertebrates and complex vertebrates. In modern medicine, researchers study their genetics and embryonic development to understand human birth defects related to the spinal cord, such as spina bifida. Beyond the laboratory, they play a vital ecological role. As prolific filter feeders, they constantly clean the ocean water. By consuming microscopic plankton, they transfer vital energy up the food web, making it available to larger predators. They are tiny, but mighty. To understand how these primitive animals survive without complex hunting appendages, we can look at the mathematics of their filter feeding mechanism. This formula calculates their energy intake. E represents the total energy intake rate of the protochordate, measured in joules per hour. Q stands for the volumetric flow rate of water pumped through their gills, in liters per hour. C represents the energy concentration of the microscopic food particles in that water, in joules per liter. Finally, the Greek letter eta represents the extraction efficiency of the animal's mucus net, expressed as a decimal between 0 and 1. If the water flow rate increases, or if the food concentration increases, the total energy intake will proportionally increase, allowing the animal to grow. Before we move on, let us see this formula in action with a concrete example. Imagine a marine biologist is studying a sea squirt, a type of tunicate. We are given that it pumps water at a rate of 2.5 liters per hour. The surrounding water has a plankton energy concentration of 40 joules per liter. The animal's extraction efficiency is 80%. First, we must convert that percentage into a decimal, giving us an eta of 0.80. Next, we substitute these values into our equation. Multiplying the flow rate of 2.5 by the concentration of 40 tells us that 100 joules of potential food energy passes through the animal every hour. Finally, applying the 0.80 efficiency multiplier, we find our final answer. The tunicate successfully harvests 80 joules of usable energy every hour to fuel its metabolism. Now that we have used this formula to find energy, let us consider a different scenario. What if we know how much energy the animal needs and we want to find out how much water it must pump to survive? We need to rearrange our equation to solve for Q. We start with our original equation, E equals Q times C times eta. To isolate Q, we divide both sides of the equation by the product of the food concentration, C, and the efficiency, eta. The terms cancel out on the right side, leaving Q completely by itself. This rearranged formula tells us that the required pumping rate is directly proportional to the energy demand and inversely proportional to both food availability and filtering efficiency. Let us apply our newly derived formula to a lancelet burrowed in coastal sand. We are given that this lancelet requires 120 joules per hour to survive. However, its habitat is nutrient poor, with a food concentration of only 15 joules per liter. Its extraction efficiency is an impressive 90%, or 0.90. We want to find the required pumping rate, Q. We substitute our known values into the rearranged equation. In the denominator, we multiply the concentration of 15 by the efficiency of 0.90, which gives us 13.5. This means the lancelet extracts 13.5 joules from every single liter of water. Dividing the required 120 joules by 13.5 yields our final answer. The lancelet must continuously pump approximately 8.89 liters of water every single hour just to avoid starvation. This calculation we just performed leads us to an important biological realization. Pumping nearly 9 liters of water an hour through a tiny body is a massive physical challenge. It perfectly illustrates the difficulties protochordates face when living in nutrient-poor waters. Because they rely entirely on the density of plankton in their immediate environment, a drop in food concentration means they must expend significantly more energy just to break even. This mathematical reality explains exactly why these animals evolved highly specialized, continuously beating cilia in their pharynx. These microscopic hair-like structures work tirelessly to maintain a constant, life-sustaining water flow. We have covered a lot of ground today, so let us summarize the key takeaways. First, remember that protochordates are the essential evolutionary stepping stones, bridging the gap between simple invertebrates and complex vertebrates. Second, even though they lack a true skull and bony skeleton, they are defined by 4 chordate hallmarks: a notochord, a dorsal hollow nerve cord, pharyngeal gill slits, and a postanal tail. Finally, as we saw through our mathematical models, mastering the art of filter feeding has allowed these humble marine creatures to survive for hundreds of millions of years. Understanding their biology gives you the ultimate foundation for studying all higher animal diversity. Great job today, and keep exploring.